In this video tutorial, I am going to discuss about pyruvate dehydrogenase complex. This pyruvate dehydrogenase complex, it catalyzes the conversion of pyruvate to acetyl coenzyme A. Acetyl coenzyme A. Now, this conversion reaction, it is of the oxidative type, and along with this oxidation of pyruvate, this carbon dioxide, one molecule of carbon dioxide is removed from the pyruvate. So, there is a decarboxylation also. So, to represent oxidation as well as decarboxylation, this process, it is also called as a oxidative decarboxylation. The second thing about this reaction is that this once acetyl coenzyme is formed, it cannot go back to the pyruvate synthesis. Okay, there is no any enzyme which can convert acetyl coenzyme A to the pyruvate. So, this is absolutely one-way pathway. Now, once acetyl coenzyme A is formed, it can have a several different fate. It can go for the fatty acid synthesis, it can go for the cholesterol synthesis, it can go for the ketone body synthesis, or it can go to the TCA cycle. Now, TCA cycle is very different from rest 3 pathway. This TCA cycle, it can generate ATPs, right? And when we want to derive energy from the carbohydrate, first we have to carry out glycolysis, then as an end product of glycolysis, pyruvate is generated. So, as an end product of aerobic glycolysis, we get a pyruvate. This pyruvate gets converted to acetyl coenzyme A, and for the energy purpose, it has to go to the TCA cycle, right? So, we can say that this reaction is a linking reaction between glycolysis and TCA cycle, right? So, now next we will discuss about pyruvate dehydrogenase complex. This enzyme is very interesting one. And in this diagram, I tried to represent schematically pyruvate dehydrogenase complex. Okay, pyruvate dehydrogenase complex, it is a multi-enzyme complex. Multi-enzyme complex. Now, why it is called as a multi-enzyme complex? The reason is that this pyruvate dehydrogenase complex, it has a multiple different enzyme activity. Actually speaking, it has 3 different enzyme activities. Okay, in this diagram, see these 3 big balloons are shown. They represent 3 different enzyme activity. The first one, that is E1, that enzyme activity is the pyruvate decarboxylase, which is represented by this balloon. This middle balloon represents second enzyme activity, that is E2, and its activity is dihydrolipoyl transacetylase. And this third small balloon, it represents third enzyme activity, that is E3, and it is dihydrolipoyl dehydrogenase. So, we have E1 activity, E2 activity, and E3 activity. E1 is the pyruvate decarboxylase. Now, here in some book or some author gives this E1 name as a pyruvate dehydrogenase. But I personally prefer this pyruvate decarboxylase name. The reason is that if we say pyruvate dehydrogenase, then this complex name itself is pyruvate dehydrogenase complex, and its one component's name is also similar, then we can get confused, right? There is nothing wrong in speaking pyruvate dehydrogenase as such, but this creates a confusion. So, I am not using this word, rather I will be using pyruvate decarboxylase. The second enzyme, it is the dihydrolipoyl transacetylase. So, dihydrolipoyl transacetylase. And the third enzyme activity is the dihydrolipoyl dehydrogenase, that is dihydrolipoyl dehydrogenase. Now, in addition to these 3 different enzyme activity, this wall complex, it requires 5 different coenzymes. Okay, and all these coenzymes, they are shown in this diagram. You can see this thiamine pyrophosphate, this lipoamide, this coenzyme A, this flavin adenine dinucleotide, and this NAD+. So, we have 5 different coenzymes. Let's discuss about each of these coenzymes. The first one is thiamine pyrophosphate. Now, this thiamine pyrophosphate, it is covalently bound with this pyruvate decarboxylase, as it is shown in this diagram. So, it is covalently bound with this pyruvate decarboxylase, right? The second coenzyme is the lipoamide, that is lipoamide. Now, this lipoamide, it is also covalently bound to this enzyme complex, but here it is bound with the dihydrolipoyl transacetylase, right? Now, what is lipoamide? See, this is an enzyme, and we know that all enzymes are protein, right? So, once it is a protein, it is of course amino acid chain. Suppose this is the amino acid chain. In the amino acid, there is a lysine is present at one particular location. With this lysine, one sulfur containing fatty acid is bound, and the name of that fatty acid is lipoic acid. So, this lipoic acid is bound with the lysine part of this amino acid backbone. Now, in this lysine, there is a epsilon amino group, and with this epsilon amino group, this lipoic acid is actually bound. So, along with this amino group and lipoic acid, we call it as a lipomide. And as I told you that lipoic acid is a sulfur containing amino acid. So, this lipoic acid, it has two sulfur atoms. They are arranged like this. Now, in the native form of this enzyme, both sulfur atoms, they are bound with each other. So, it looks like a disulfide bond. And this is the oxidized form of lipoamide. And this is found in the native structure of this pyruvate dehydrogenase complex. But it can also occur in the reduced form. So, how does reduced form looks like? So, lipomide with its two sulfur atom, when both are bound with this H atom, it becomes our reduced form of the lipoamide. But remember, this reduced form, it is not found in the native structure of this pyruvate dehydrogenase complex. Okay, it is the oxidized form which is found. The third coenzyme is the coenzyme A, that is coenzyme A. Now, in this coenzyme A, there is one very important group, that is SH group, and to emphasize that SH group is important in the coenzyme A, we generally write coenzyme A with its SH group, right? Now, see, this coenzyme A, it is not bound with any of the protein component of this pyruvate dehydrogenase complex. It is lying in the free, but it is required by this enzyme E2, okay? So, that thing we have to keep in mind. The fourth coenzyme is flavin adenine dinucleotide. This flavin adenine dinucleotide, it is covalently bound with this E3 part, that is dihydrolipoyl dehydrogenase. And the last and final coenzyme is this NAD+, that is nicotinamide adenine dinucleotide. It is just like this coenzyme A, this NAD is not bound with this any of the protein part, it is free. So, coenzyme A is free and NAD, it is also free, but they are required for this activity, proper activity of this complex. This NAD is required for the proper activity of E3 and coenzyme A is required for the proper activity of this E2 part. That thing we have to keep in mind. One more thing about this coenzyme is that all of this coenzyme, they are more or less derived from different vitamins. See, thiamine pyrophosphate, it is derived from the vitamin thiamine. You can also call it as a vitamin B1. This flavin adenine dinucleotide, it is derived from the riboflavin. We call it as a vitamin B2. This nicotinamide adenine dinucleotide, it is derived from the niacin, that is vitamin B3. Right? This coenzyme A, it is derived from the vitamin pantothenic acid. And rather to consider this lipoamide as a product of vitamin, it is debatable. See, some author considers lipoic acid as a vitamin and some author does not consider it as a vitamin. So, this is a debatable topic. So, I will leave up to you that whether you consider it as a derivative of vitamin or not. So, this is more or less a structure of pyruvate dehydrogenase complex. Now, one more thing about that I had omitted one thing from this diagram. And that is that in this complex I had shown only one copy of pyruvate decarboxylase, only one copy of dihydrolipoyl transacetylase and only one copy of this dihydrolipoyl dehydrogenase. But in reality, there are multiple copies of pyruvate decarboxylase, multiple copies of this each different enzyme. Okay, so because of multiple copies of particular enzymes are present, we can consider it as a cluster or aggregate of enzyme having a multiple copies of each enzyme. See, here we come across two words. one is multi-enzyme complex. Why we call it as a multi-enzyme complex? Because it has a multiple different enzyme activities. And second term, that is why we call it as a cluster or aggregate of enzyme, because each enzyme activity is, there is a multiple copies of this, each different enzyme are present. So pyruvate dehydrogenase complex is a very big molecule. See, just to give example, this pyruvate decarboxylation, it is present in 20 to 30 copies. This dihydrolipoyl transacetylase, it is present in approximately 60 copies of this enzyme is present in one single complex. And regarding this dihydrolipoyl dehydrogenase, approximately 20 to 30 copies are present in single pyruvate dehydrogenase complex. So this number of copies, it is different for the different species, but indeed there is a multiple copies present in one single enzyme, right? So now let us look at the mechanism of action of this pyruvate dehydrogenase complex. And for that also we need a diagram of this pyruvate dehydrogenase complex, right? See, this pyruvate dehydrogenase complex, it is located in the mitochondria, right? And in the mitochondria, especially it is associated with inner mitochondrial membrane. Now, what this pyruvate dehydrogenase complex does? It converts pyruvate to the acetyl coenzyme A, right? But the problem is that this pyruvate, it is produced in the cytoplasm. So now if this pyruvate needs to undergo oxidative decarboxylation to give rise to acetyl coenzyme A, it has to first transported inside the mitochondria. So from the cytoplasm, first pyruvate is transported across this mitochondrial membrane, and now pyruvate is inside the mitochondria. This transport is carried out by one protein transporter present in this mitochondrial membrane, and its name is proton symporter. Proton symporter transports pyruvate from the cytoplasm to the mitochondria, right? Now, once pyruvate is inside the mitochondria, it can react with this pyruvate dehydrogenase complex. So, first, pyruvate is acted upon by this pyruvate decarboxylase. So, what this pyruvate decarboxylase does? Pyruvate decarboxylase, it removes one carbon in the form of carbon dioxide from the pyruvate, right? So, in this pyruvate, there are total 3 carbon, and 1 carbon is removed in the form of carbon dioxide. So, the remaining carbon skeletal, it is now called as a hydroxyethyl, and that hydroxyethyl compound, it is transferred to this TPP coenzyme of this pyruvate decarboxylase. So, now instead of TPP, we will have a hydroxyethyl TPP, hydroxyethyl thiamine pyrophosphate. Now, this hydroxyethyl thiamine pyrophosphate, it needs to lose this hydroxyethyl and convert back to its original form. So, that reaction is catalyzed by this dihydrolipoyl transacetylase. So, now this thiamine pyrophosphate is getting regenerated, right? Then what happens to this hydroxyethyl? This hydroxyethyl is handed over to this lipoamide coenzyme of this dihydrolipoyl transacetylase. See, this lipoamide, it has 2 sulfur atoms, right? So, from this hydroxyethyl, one hydrogen is given to this lower sulfur, and remaining carbon skeleton, now it is known as acetyl. So, it is given to this second sulfur. So, now this is a structure of lipoamide and this is acetyl. So, now this compound, it is known as acetyl lipoamide, right? So, simultaneously it is converting into this form, and this reaction is catalyzed by this E2. Right, and this reaction was catalyzed by this pyruvate decarboxylase, that is E1. Now, what happens to this acetyl lipoamide? See, this acetyl group is then transferred to this coenzyme A. Coenzyme A has this thiol group, and with this sulfur, acetyl group get attached. So, this coenzyme A, it will now be like looking like this coenzyme A with its sulfur, and with this sulfur, acetyl group is attached. Now, this thing is nothing but our acetyl coenzyme A. Okay, this structure, it is known as acetyl coenzyme A, and simultaneously this hydrogen is given to this sulfur. So, now our lipoamide, it looks like L, and here SH and here SH. So, see, this is the structure of lipoamide, but whether it is oxidized or reduced, it is a reduced form of lipoamide, right? And this reaction is also catalyzed by the same enzyme, that is E2. Now again, this reduced form of lipoamide, it needs to get converted to oxidized form, right? Because otherwise this enzyme cannot attain its native form. So this 2 hydrogen needs to get removed. So that 2 hydrogen is removed by this dihydrolipoyl dehydrogenase. What this FAD does? It takes up this both hydrogen, so it converts to FADH2. And by losing both hydrogen, lipoamide is regenerated in the oxidized form, whereas FAD is now reduced to this FADH2. Now this FADH2 also needs to convert back to its native form, to FAD. So what happens? This NAD plus, it takes up this 2 hydrogen and itself get convert into NADH plus H plus. So this completes the mechanism of action of pyruvate dehydrogenase complex. As you can see that here we have total 3 substrate. One is this pyruvate, second one is this coenzyme A, and third one is NAD plus. And here we have 3 different products. These products are, one is carbon dioxide, second one is acetyl coenzyme A, and third one is NADH and H plus. So if I want to write down this entire reaction in the simplified form, I can write it like this. We have a 3 substrate, right? So we have a pyruvate plus coenzyme A plus NAD plus. And we have a 3 product, that is acetyl coenzyme A, then we have NADH plus H plus, and then we have a carbon dioxide, right? And this entire reaction is catalyzed by this PDH complex. So here you can see that acetyl coenzyme A, it will further travel to the TCA cycle, and this NADH plus H plus, it can generate ATP, right? So it can generate ATP by the electron transport chain. And we know that each molecule of NADH, it has a potential to generate 2.5 ATPs. We also know that one molecule of glucose can give rise to 2 molecules of pyruvate. So we can say that by this oxidative decarboxylation, one molecule of glucose generate 2.5 ATP 2 times, that makes total 5 ATPs, right? Now next we need to see about the regulation of pyruvate dehydrogenase complex. So pyruvate dehydrogenase complex, it is regulated by 2 different ways. One way is the end product inhibition. End product inhibition. And second way is the covalent modification of enzyme. Covalent modification. Let's first look at this end product inhibition. Remember, whenever end product inhibition is there, most of the time it is allosteric regulation, right? So in this case, in this entire reaction, we know that acetyl coenzyme A, NADH and H plus, and carbon dioxide, these 3 are the product. So out of these 3 products, acetyl coenzyme A and NADH and H plus both are allosteric inactivator of this pyruvate dehydrogenase complex, and the specific location in the PDH complex is the E1 part. Both of these act on the E1 part as an allosteric inhibitor, and so this PDH activity can be kept under check. The second way of regulation is the covalent modification. See, pyruvate dehydrogenase, it can occur in the 2 form. One, PDH form like this, where there is no any phosphate, and second one, PDH, where it is attached with the phosphate. We call this compound as a phosphorylated PDH, whereas we call this compound where we do not have phosphate, we call it as a dephosphorylated. And dephosphorylated form of pyruvate dehydrogenase complex, it is actually the active compound, whereas the phosphorylated one is the inactive. Now PDH can be phosphorylated by the enzyme PDH kinase. What this PDH kinase does, it transfers phosphate from this ATP, it gives to this pyruvate dehydrogenase, and ATP gets converted to ADP, right? And this phosphate can be removed, or it can be dephosphorylated by the enzyme PDH phosphatase. PDH phosphatase removes this phosphate as an inorganic phosphate. So, inorganic phosphate is removed, but water molecule is added. So, that is a hydrolysis. Okay. Now, this PDH kinase, it can get stimulated by the presence of acetyl coenzyme A or NAD or ATP, right. All these three are the indicator of energy rich state. So, what will happen, this energy rich indicator acetyl coenzyme A, NADH and ATP, these are the energy rich indicator, they stimulate PDH kinase and thereby they inactivate this enzyme. So, they will make sure that no more pyruvate gets converted to acetyl CoA, right, because now the oxidation is not required as already we are in the energy rich state. Whereas the counterpart of this each three, that is counterpart of acetyl CoA is coenzyme A, counterpart of this NADH is NAD plus and counterpart of ATP is the AMP. These three are the energy poor indicator. They indicate that cell is now under energy poor state. So, what they do? They inhibit this. So, what will happen? PDH will remain active and it will generate more and more ATPs, right. So, see how well it is regulated. Now, actually speaking, this energy rich and energy poor indicator, it is actually a ratio. See, if you look at the ratio of acetyl coenzyme A to coenzyme A, or you can look at the ratio of NADH to NAD plus, or a ratio of ATP to AMP. So, these three are basically a cell's energy status. Okay, if acetyl coenzyme, this ratio is increased, this ratio is increased, and this ratio is increased, that means cell is currently in the energy rich state. If all these three ratios are decreased, they indicate that cell is now in the energy poor state, right. So, this ratio are actually important than these individual compounds. Now, PDH phosphatase is also under regulation. See, insulin stimulates this PDH phosphatase and this occurs specially in the fat tissue or adipose tissue. So, what will happen in the presence of insulin? More of the PDH will be active. So, more and more acetyl coenzyme A will be formed. And this more and more acetyl coenzyme A, it can be converted to fat, cholesterol or ketone bodies, right. So, that is the function of insulin. The second interesting aspect is that this calcium ion, they have also potency to stimulate this pyruvate dehydrogenase phosphatase, and by stimulating it keeps PDH in the active form. Now, what should be the purpose of this calcium stimulating this PDH phosphatase? The answer is that skeletal muscle, when they undergo contraction, at that time calcium is released. So, this released calcium will make sure that enough of the PDH activity is there, and by that enough of the ATP will be generated, right. So, when skeletal muscle contracts, they need energy in the form of ATP. So, that is carried out by this calcium ion by stimulating PDH phosphatase. So, this is the regulation of pyruvate dehydrogenase complex. Now, next we will see biomedical significance or biomedical importance. What is the biomedical importance of this pyruvate dehydrogenase? The first and most common thing is thiamine deficiency. Thiamine, that is vitamin B1 deficiency. See, if thiamine is deficient, what will happen? There will be low activity of its coenzyme form, that is thiamine pyrophosphate, right. And if this coenzyme is not working, that means pyruvate dehydrogenase will not work. Now, we know that pyruvate dehydrogenase, it is required for the conversion of this pyruvate to acetyl CoA, and it also generates ATP, right. So, if this reaction is not taking place, what will happen? There will be increased concentration of pyruvate, and now this increased pyruvate, it gets diverted to formation of lactic acid. So, obviously lactic acid concentration will also increase, that result into lactic acidosis. So, lactic acidosis is one of the obvious symptom of this thiamine deficiency. Now, second thing occurs that there is no any supply of enough ATP, right. Now, in our body there are certain tissue which is totally dependent on the carbohydrate for the energy purpose. One of the tissue is brain. Now, brain generally derive energy from where? From the glycolysis, from this, then from this oxidative decarboxylation and from the tricarboxylic acid cycle. So, if in the brain what will happen? this step will not occur. So there will be a less supply of ATP, right? If carbohydrate needs to generate energy, it has to pass through this pathway, but that is not happening in the deficiency of thiamine. So there will be a less ATP. So it is a damaging thing. Less ATP is the damaging thing for the neuron cell, and in this acidosis will add to the damage to the brain. So there will be a neurodegenerative changes. Neurodegenerative changes will be observed in case of thiamine deficiency. The second clinical significance is that rarely PDH enzyme is deficient. PDH enzyme is deficient, and especially its E1 part, there is a mutation in the gene which encodes some part of this enzyme E1, that is pyruvate decarboxylase, right? So what will happen to this pyruvate dehydrogenase enzyme deficiency? The same thing, lactic acidosis, and it will lead to neurodegeneration. The same effect you will see, and this is one of the cause of congenital lactic acidosis. This congenital lactic acidosis, it is one of the very rare disease, okay, but although it is a rare disease, its most common cause of this congenital lactic acidosis is the pyruvate dehydrogenase enzyme deficiency. So here what will be the treatment? As such, there is no any definitive treatment, but yes, we can symptomatically treat this patient. In such patient, if we give mega dose of thiamine or vitamin B1, then it is seen to some improvement in such child. And the second thing we need to do in such patient is that there should be dietary restriction of carbohydrate. Carbohydrate should not be given in the diet. Okay, if we don't give carbohydrate, then there will be less action will be required from this pyruvate dehydrogenase enzyme. The third important biomedical importance is the arsenic poisoning. Arsenic poisoning. Now, arsenic occurs into its trivalent form. Arsenic ion occurs actually in its trivalent form, and it is known as arsenite. This arsenite, they inhibit pyruvate dehydrogenase complex. Okay, so what will happen if pyruvate dehydrogenase is not working? The same thing, lactic acidosis and neurodegenerative changes, this thing occur. Here I would like to refresh your memory that earlier also, while discussing glycolysis, we had talked about arsenic poisoning. In that glycolysis, we had talked about its pentavalent form. Its pentavalent form that we call as a arsenate. Arsenate. This arsenate, it cannot inhibit pyruvate dehydrogenase complex, rather it inhibits glyceraldehyde 3-phosphate dehydrogenase enzyme. So this distinction we have to remember. Arsenate inhibits enzyme of glycolysis, whereas arsenite it inhibits this pyruvate dehydrogenase complex. So that's all about pyruvate dehydrogenase complex. If you have any query or confusion, please write it down in the comment section below. Thank you.